hi, a very happy good evening. So this is Krishnaveni and we'll be talking about the story of life on earth, and that's biology. So welcome to my session. So today it's again a combination of chapters. So we are going to discuss 3 chapters from plant physiology. Well, for all those who are new to my lecture, so Monday, Wednesday, Friday I teach a class 12 chapter. Tuesday, Thursday, Saturday I teach a class 11 chapter. So Anusha, hello, hi 𝐎𝐩𝐿𝐿. So thank you so much for joining my lecture. So please don't forget to like, share and subscribe. Okay, so again we are going to discuss a combo of chapters today. So we are going to discuss transport in plants, mineral nutrition and plant growth development. So I have done photosynthesis and respiration separately because those 2 chapters have a lot of confusion with the ATP requirement, the amount of NADH turned over, etc. So these 3 chapters are a bit boring if taken separately, right? So you guys don't feel like listening. So now what I did was I have 10 questions from each of the chapter. I have 10 questions from transport in plants, 10 from mineral nutrition and 10 from plant growth development. Okay, so we will discuss each of the question, and with respect to the question, if there's some theory that I want you to remember, I will teach you that. Okay, so in this way we will revise all the 3 chapters. So we will be done with class 11 portion today. So we'll try our best to finish off all the 3 chapters today. Okay, hi Wave, yes, welcome to my lecture. So thank you so much for watching my lecture. So please do like, share and subscribe. Okay, and yesterday evening when we were discussing microbes in human welfare, so there was a technical glitch, so my video was not pretty clear. So I will continue microbes in human welfare on Monday along with biotechnology 1. So I will discuss the remaining questions there. Hi Antonia B, yes, a very good evening. So welcome to my lecture. Yes, I'm good. How are you? So thank you so much guys for joining my lecture. So please do like, share and subscribe. So here let's talk about the 3 of the tricky but important but boring chapter. Okay, so we have a lot of ifs and buts in plant physiology. If it's human physiology, we have no doubt, so we start and we finish off, right? But these are some chapters which we need external motivation, because internal motivation does not come, because anyways we are not strong with the concept, right? So don't worry, so we'll have a quick glance of this. But in transport in plants, there are certain terms which I will recap once again, and then we will start solving the questions. Okay, so first we start with transport in plants. So in transport in plants, we spoke about the means of transport, active transport, passive transport. So in passive, we have 2 types. We have diffusion and facilitated diffusion. In facilitated diffusion, we have 3 types: symport, antiport and uniport. So please look into the examples of these 3. Then we went on to talk about what is plant-water relation. So we have this term. So water potential is equal to solute potential plus pressure potential. Okay, so this is the term that we are supposed to discuss. So your water potential for pure water is maximum, that is 0. Thus a water potential for a solution is negative. So solute potential is always negative and pressure potential is always positive. So these are some of the basic criteria that you should know. Right, so how for long distance water is transported? You have your cohesion, adhesion, transmission, pull theory. So you have to remember that. So then we differentiated what is transpiration and what is photosynthesis, and how does uptake and transport of mineral nutrients happen. Finally, what is phloem transport, right? So apart from that, here you have certain concepts which we are going to discuss. So I told you water potential. Yes, so for pure water, water potential is 0. So this is for pure water. So thus for a solution, water potential will always be negative, right? So pure water has more water molecules compared to a solution. So solute potential is psi s. So solute potential is always negative. So this is pressure potential or turgor pressure, which is always positive. So thus the sum of the equation is water potential is equal to solute potential plus pressure potential, right? Okay. The next one is what is diffusion pressure? So that is dP. So diffusion pressure is the ability of a molecule to diffuse from a higher concentration to a lower concentration. That is diffusion pressure. So diffusion pressure is an ability of a molecule to diffuse from a higher concentration to lower concentration. So all molecules cannot diffuse. So water diffuses because water has a definite diffusion pressure, right? So what is DPD? That is diffusion pressure deficient, or it is also known as your suction pressure. So what is this DPD and what is this suction pressure? So this is the difference in the diffusion pressure. So this talks about the difference in the diffusion pressure of water in its pure state to the diffusion pressure of water in solution. So this is your DPD. So DPD is equal to osmotic pressure minus your turgor pressure. So now what is osmotic pressure? So the pressure exerted by the solute molecules in a solution to prevent the entry of water inside. Suppose if I have pure water here, okay, so this is pure water, so I have a solution. A solution is a combination of solute plus solvent, right? So solvent here is my water and solute here is my sugar. So now by the law of universe, so water has diffusing potential, so water will move inside, right? So this is where you have high concentration of water and here you have low concentration of water. So water will move here, right? But this solute molecule will resist this flow of water because more water comes, the solution will become diluted, right? So the pressure exerted by the solvent molecule to stop the flow of water into the solution, so that is your osmotic potential. Yes? Yes? Okay. So here, in case of diffusion, so water moves from higher concentration to lower concentration, right? So when you are talking about water movement with respect to DPD, so DPD moves from lower DPD to higher DPD. So a region with DPD, okay, so when I say lower DPD, the difference is less. So that's the water, so which means the water is maximum here. The solute molecules are less and water is maximum. So it flows from here to here. Alright, so all these terms are clear? Yes? So this is how it defines the water movement. Okay, Anthony Abhi? Fine. So osmotic pressure I have explained. So moving further, so water potential is also explained. So now apart from this, you have certain other conditions like this, plant cell in osmosis. So what is all this we will talk about? So here I am talking about your hypertonic solution. Hypertonic solution. So hyper is high, tonic is high ionic concentration, so low water concentration. If my cell is placed in a hypertonic condition, so which has more amount of ions and less water, okay, so osmotic potential is negative, osmotic pressure is positive. So osmotic pressure is equal to osmotic potential. So this is positive, this is negative. So this is potential. Okay? Yes? Fine. So now what happens? Water from the cell moves outside. So water moves out of the cell. So thus the cell shrinks. So when the cell shrinks away from the cell wall, so such a cell is known as your plasmalized cell. Okay? Fine. So now I am talking about hypotonic condition. So when your cell is placed in a hypotonic condition, right? So which means I have less of ions and more of water. So water from the surrounding medium moves into the cell. So water moves into the cell, right? So when water moves into the cell swells up, right? So such a cell is known as your turgid cell. Clear? Okay. So the third condition is isotonic. So when I place my cell in an isotonic means same concentration of ions and same concentration of water. So here there is no net movement of water, right? So the cell remains normal. So such a kind of cell is known as your flaccid cell. Okay. So here in your flaccid cell there is no movement of water. So your turgor pressure is 0. Okay. So here your turgor pressure is maximum. So this is basic logic, right? Yes, Darni, a very happy good evening. So thank you for joining my session. So please do like, share and subscribe. Okay. So we have hypertonic, hypotonic and isotonic. Right. So the same thing is mentioned here. Okay. So moving forward, so dPd is equal to OP minus turgor pressure. Right. So if the wall pressure is equal to turgor pressure, so sometimes this equation is reframed as osmotic pressure minus wall pressure. The next one is a flaccid cell. So in a flaccid cell there is no net movement of water. So turgor pressure is 0. Thus your dPd is equal to osmotic pressure. So this is understood, right? Solute particles increase solute potential. Solute particle increases, solute potential decreases. Yes, because more the number of solute particles, the negative the value of solute potential becomes. So if I have more solute potential, right, if I have more solute molecules, it can be minus 10, it can be minus 100, right? So the value keeps on decreasing in the negative scale when you have more amount of solute particles. Yes, Vishal, hello, very happy good evening. So thank you for joining my session. So please do like, share and subscribe. Okay. So in a turgid cell, water becomes turgid and the cell membrane expands due to the extra intake. So this is when it is placed in a hypertonic solution, right? So here your dPd is 0 because your osmotic pressure is equal to your turgor pressure in your turgid cell. So thus your dPd is 0 in your turgid cell. So thus no water enters inside, right? So it is already full. There is no space. The cell is swelled up. So here your turgor pressure is equal to your osmotic pressure. So plasmalized cell, your dPd is maximum. Why? In plasmalized cell, because your turgor pressure is negative. A plasmalized cell is when it is placed in a hypertonic solution. So this is placed in a hypertonic solution. So in hypertonic we have more ions and less of water. So water moves out of the cell, right? So the cell will shrink. So if the cell shrinks, there is no pressure on the walls, right? So your turgor pressure becomes negative. So if it's OP minus TP is equal to dPd, if this is also in your negative value, so then the value will become maximum. So here your TPD is maximum. So dPd is maximum here. Okay. So you have to remember these logical correlations so that it is easy. Fine. So then please remember what is guttation. So guttation is the liquid loss of water, right? Okay. In hypotonic, dPd is equal to, no, in hypotonic, okay, so how do you understand? So in hypotonic, what happens? So you have more of ions, you have less of you have less of ions and you have more of water. So water moves inside the cell. So when water moves inside the cell, the turgor pressure and the osmotic pressure is equal. So in your hypotonic condition, dPd is 0. So hypotonic condition is a turgid cell. Okay, Vishal? Yes, you got the logic? Fine. So apart from this, you should know what is guttation, you should know what is imbibition. So imbibition is the absorption of water by solids. Okay. The next thing that you should know is phloem transport. So how does it happen? So when we were talking about phloem transport, so we spoke about what is a source and what is a sink. So source is the place where food is present and sink is the place where food has to be transported. So this source and sink can be interchanged because that is the reason your phloem is bidirectional. Okay, right. So suppose the food that I have, yes, so food that the glucose, by the process of photosynthesis, it produces glucose. So glucose is converted into starch. Okay, Okay, YouTube only invited you for 5 lines, okay, so when you continuously type without any gap, so they told me that is when it times out you, maybe that is because of that, I am not sure, okay. Hi Vinay, yes, welcome to my lecture, so thank you so much for watching my lecture, so please do like, share and subscribe, okay. So this moves into the phloem tube, so this moves by active transport, okay. So when the food particle moves into the phloem tube, right, so what happens, this becomes hypertonic, so which means it has more ions and less of water. So next to the phloem you have the xylem, so thus the water from the xylem enters the phloem, right. And now this moves down to the sink and then it is unloaded. So you should remember here the solution becomes hypertonic. Fine? Yes, answer this question for me. In a fully turgid cell, so in a fully turgid cell, which means it is hypotonic. So solute molecules, the solute potential will be negative and pressure potential will be positive. So solute potential will be negative and pressure potential will be positive. Both will be positive, both will be negative. So what will happen here? What will happen here? So it's a simple question. In a fully turgid cell, that is when the cell is placed in a hypertonic condition. So what will happen? Yes, come on quick. The quicker we answer, so we can finish off all the 3 chapters today. Yes, come on. So what will happen here? So what will happen? So in a hypertonic solution, dPd will be equal to 0. So here your osmotic potential will be your turgor potential, right? Yes. But in a turgid cell also you have water potential is equal to solute potential plus pressure potential. So from given terms, right, so here it will be solute potential will be negative and your pressure potential will be positive. If both are negative, then dPd cannot be 0. If both are negative, dPd cannot be 0. So here the right answer is option A. Hi Kathy, hello, how are you? So thank you for joining my lecture. So please do like, share and subscribe. So here option A is the right answer. So question 2. Phenomenon of plasmolysis occurs when cells are kept in a hypertonic solution, cells are kept in a hypotonic solution, cells are kept in a hypotonic solution. Both are the same. Okay, so anyways BC is not the answer. So here's A is the answer. Right? So plasmolysis occurs when it is present, when it is kept in a hypertonic solution. When a cell is plasmolysed, it becomes flaccid and turgor pressure becomes 0. Turgid and the turgor pressure becomes 0. Turgid and the turgor pressure becomes equal to your osmotic pressure. So what happens here? So question number 3. So when a cell is plasmolysed, okay, so this also happens, okay, fine. So when the cell is plasmolysed, so your turgor pressure becomes negative, right? So your dPd is, that is why your dPd is maximum in a plasmolysed cell. So in a plasmolysed cell, the cell is, so it is neither flaccid, it is neither turgid. Okay, so here we can also consider it to be 0, so this is negligible, right? So here we call it as flaccid and the turgor pressure becomes 0. So if the turgor pressure is equivalent to 0, then the cell is flaccid cell. If the turgor pressure is negative, so both are the same, it is never positive, right? So this is the most closest answer. You're getting? Yes? Fine. So question 4, seed increases in its volume by the absorption of water through osmosis, diffusion, imbibition, plasmolysis. So here what is the answer? So here what is the answer? So seed increase in its volume by the absorption of water through osmosis, diffusion, imbibition, plasmolysis. So this is imbibition, right? So question 5, a pressure that is responsible for pushing up water to a small height in the stem is called as positive root pressure, turgor pressure, pressure gradient, negative root pressure. Yes, what is the right answer here? So it is positive root pressure, right? So root pressure is always positive. So here the answer is A. So moving forward, guard cells help in protection against grazing, transpiration, guttation, fighting against infection. So what is the function of the guard cell? Question number 6. So this is transpiration, right? So this is option B. Done? Okay. So moving further. So question number 7. So sinks are related to the transport of minerals, stomata, hydrothotes or phytochromes. So sink and source are related with the transport of minerals. So these are pretty easy questions. So question number 8. So translocation of food in the flowering plants occurs in the form of starch, glyceraldehyde, glucose, sucrose. Yes, question number 8. So I guess I told you wrong. So it is not, so photosynthesis produces glucose, but it is not transported in the form of starch, but it is transported in the form of sucrose. Sorry, in the previous slide I wrote it as starch, but the right answer here is sucrose. Okay? Yeah. So question 9, dash is mainly water and sucrose, but the other sugars, hormones and amino acids are dashed through the phloem. So which of the fill in the blanks is right here? Which of the fill in the blanks is right here? So phloem sap is mainly water and sucrose, but other sugars, hormones and amino acids are also translocated through the phloem. So this is option B, right? So question number 9 is option B. So this is your phloem sap. And these are also translocated. So this is the answer. Okay? Yes, fine. So question 10, the hypothesis accepted for the translocation of sugar from the source to the sink is which one? Pressure gradient, pressure flow hypothesis, mass flow hypothesis, both B and C. So which is the most accepted hypothesis? So it is both pressure flow and mass flow. So the right answer is option D, right? 10 is C, no, pressure flow is also known as mass flow. So with this we are done revising first chapter, that is transport in plants. Now we move to the second chapter, that is mineral nutrition. So in mineral nutrition, what are the tough things? So in mineral nutrition, ma'am, we have the micronutrients and macronutrients. Listen, things can be sorted, so nothing is very complicated. So in your mineral nutrition, you have to remember the criteria. Criteria as to why a particular nutrient is a essential one, right? So the first criteria is that it should be really needed for the life cycle, right? So first criteria is that it should be absolutely necessary. It should be the important essential nutrient. Then it should not be replaceable. And it should be involved in metabolism, right? So then we have divided nutrients into macro and micronutrients, right? So macronutrient is excess of 10 millimole per kg dry matter. So this is less than 10 millimole per kg dry matter. So that is why this is macro and this is micro, right? So totally we have 8 plus 5. I guess we have, yeah. Okay, so let's see what are the nutrients that are important. Okay, so first we have your potassium, phosphorus, potassium, nitrogen, sulfur and boron. So these are single symbol, right? So you have your potassium, phosphorus, potassium, nitrogen, sulfur, boron. Then you have your calcium, chlorine, and then copper, right? So you have your iron, you have magnesium, you have manganese, you have molybdenum, and then you have zinc, right? So it is 8 plus 5, so we have totally 14 elements which we are going to study in detail, right? So how do you remember them? So we will start from here, okay? So first we are talking about your nitrogen. So nitrogen is most abundant, okay? So that is the reason I am talking about nitrogen first. So when we know something well, we solve that first, right? So nitrogen is most abundantly present. So remember that. And always it is N2. We always need it in more number. If I say if this is for 100, you always want, so if mummy says she is going to give you 100, you will say, mummy, please give me 110, right? So now nitrogen is uptaken in the NO3 form or in the NH4 plus form. So it is normally in N2. So we take it up in 3 or the 4 ionization form. So remember it like that. So where our nitrogen is present? So it is Mn, right? In alphabetical order we have M, then we have N. So where is nitrogen present? So nitrogen is present in metabolically active tissues. It is present in metabolically active tissues as well as in again M, that is meristematic tissues. So M is always present, N is always present with M. So it is present in metabolically active tissues and meristematic tissues, okay? So that is about nitrogen. So now let's talk about your phosphorus. So phosphorus, how it is present? So phosphorus is taken in H2PO4 minus or HPO4 2 minus. Okay, so we know phosphorus is a constituent of the cell membrane. So we have phospholipid bilayer. So it is a constituent of your nucleic acid because all of them are made up of nucleotides. Okay, so these are the 2 points in phosphorus. So we are done with phosphorus now. So the next one, let's talk about your potassium. So potassium is a very simple fellow. It is taken in the K plus form. Okay, so what is the function of potassium? It helps in maintaining the cationic anionic balance. So it helps in the opening and closing of stomata. So if it helps in the opening and closing of stomata, it helps in maintaining the turgidity of the guard cells. And to some extent, it also activates enzyme, but what enzyme is not mentioned, so we are not going deep. Fine. So we are done with potassium. Now coming to sulfur. So sulfur is very easy. So why sulfur is very easy? Because sulfur, the functions are easy. So sulfur is taken in SO4 2 minus sulfate form, right? H2SO4 we study, no? Fine. So where are sulfur present? So we know amino acids which has sulfur, right? So that is your cysteine and methionine. So apart from that, so sulfur is also present in a lot of enzymes, coenzymes, as well as in iron ferridoxin complexes. It is also present in a lot of vitamins. So what are the vitamins? So vitamin B1, that is thiamine. So again B, biotin, and then it is present in your coenzyme A. So very easy, directly we are talking about the constituents of sulfur. Fine. So with this we are done here. Okay, so these are the things we are talking about in minerals. Okay, so I don't think I left any space, that's fine. That's completely fine. So let's continue. So now we are supposed to talk about the 2-lettered symbols, right? So we have calcium, we have copper, we have chlorine. So calcium is the oldest mineral. Remember it like that. Okay. So calcium is present in your meristematic tissues. Okay. So where else is calcium present? It is present in differentiating tissues. It is also present in older leaves. So calcium is the oldest element. Okay. Yes. So now calcium is present, or it has a function in the cell membrane, it has a function in the mitotic spindle, so everything with M, and it has a function in the middle lamella. So in middle lamella we have calcium pectate, right? Okay. So apart from this, what are the functions? So it activates enzymes to some extent, and it also plays a role in the metabolic activities. So this is about your calcium. So calcium is taken in the Cu2 plus, okay. So the most easiest one is copper. So Cu2 plus, this is taken in the form of Cu2 plus, okay. So this is involved in your redox reactions, and it can be easily oxidized from Cu plus to Cu2 plus. Just 2 points, this is your copper. So now we are talking about your chlorine. So chlorine is all the more easy, so it is an anion. So it also helps in maintaining your anion-cation balance. So that is why you have a chloride shift, right? So chloride also play a role in the water splitting reaction of your photosynthesis. So when you split water, you get oxygen. So chlorine is essential for that reaction. So we are done with all the elements that is starting with C. Is it not easy? Okay. So the next thing, so it is your iron. So it is in the ferric form. So we have ferridoxin complex. So we have cytochrome complex, right? So it is also involved in the formation of your chlorophyll. So again, it is related with plant, and it helps in the activation of a particular enzyme, that is your catalase enzyme. Okay, so this is about your iron. Fine. Okay, so we are done. So now we move into your magnesium. So magnesium is used in Mg2 plus form. So what are the functions of magnesium? So we know magnesium is used in ribosome structure. So magnesium is the center part of your chlorophyll molecule. So it is present in chlorophyll. So magnesium is also a part of the enzymes involved in respiration and photosynthesis. Yes? So this is magnesium. Only 2 points in magnesium. Now how about manganese? So manganese also 2 points. So what are the points in manganese? So manganese is also Mn2 plus. Just like magnesium, it also has enzymes involved in respiration and photosynthesis of plants. Apart from that, manganese is also involved in the splitting of water during your photosynthesis. And the next element with M is molybdenum. So this is MO 4 2 minus, I guess. So this is molybdenum, if I am not wrong. No, it is 2 2 minus. Sorry, 2 2 plus. So it is always positive, molybdenum. So molybdenum is involved in your nitrogen metabolism. So it has your nitrogenase enzyme, and it also has nitrate reductase enzyme. Fine. So the last two we are left with is your zinc. So zinc is Zn2 plus. So zinc is involved in the activity of your carboxylase enzyme, and it is involved in Auxin synthesis. The last one that is left is your single syllable, is your boron. So boron is your boss. It has many functions and it is also important. So what is this boron, or what are the functions? It is taken up as BO3 minus or B4O7 2 minus. Okay, so this is the boss, right? So this is 3 minus, this is 2 minus, so it has higher numbers. Okay, so what are the functions of boron? So the first function, it is involved in pollen germination. So it is involved in the uptake and utilization of calcium. So calcium is C. It is also involved in your carbohydrate translocation. So this has quite a lot of functions. So B is the boss. So boron is the boss here. Okay. And the fourth step, it is also involved in cell differentiation. So sorry, in your cell elongation and in your cell differentiation, right? So these are the functions. So once your cell elongates, it differentiates. So boron is the boss. So this is the mechanism. So clear? Yes. So are we done with all the 14 elements very quickly? Was it tough? So remember like this, all the elements starting with M. So all the elements starting with C, then it is not tough. So you have one element that is starting with F and Z. So remember all the monosyllabic letters, so it is much easier to remember. So you got it? Yes, all of you? Any doubts in the metal ions? Yes? I hope it's clear. Done? Okay. So apart from this, in mineral nutrient, what else is there? So we have your nitrogen metabolism. So you should know the terms like what is nitrification, what is ammonification, what is denitrification, what are the bacteria that works there, that you should know, and how much of ATP, what is the final fate of your ammonia. So these points you should remember, okay? Yes. So let's go forward. So question 1, hydroponics refers to the development of the plant without soil, in saline soil, in water without soil with alkaline pH. So hydroponics is the refers to the plant development without soil, right? So this is option A. Fine? Okay. So the amount of trace element per kg matter is what? So we are talking about the trace elements. So here the trace elements. So I am sorry, so it is not without soil. So it is in water without soil. So water will be there, irrespective of the media, water will be there. So here the trace element is less than 10 millimole per kg dry weight. So here it is option 3. So dash is a constituent of the ring structure of chlorophyll and helps to maintain the ribosome structure. Abhi we saw, so it is magnesium. So this is Mg2+. Mg2+ is needed for the binding of your magnesium, right? So which of the following essential element is required for the photochemical reaction involved in the photolysis of water? So what will be your answer here? So chloride ions. So this is needed in splitting of water molecules, right? So this is Cl-. So copper, it is only used in your redox reaction. So zinc is used in the activation of carboxylase and it is used in the production of your auxin. So magnesium, it is used in the enzymes of your photosynthesis, respiration. It is used in your chlorophyll structure as well as in the structure of your ribosome. So what is left? Chloride ions. So chloride ions, apart from it is used in the photolysis of water, it also helps the anion-cation balance. Right? So it is option B. Yes sir, hello, very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So which of the following elements in plants are relatively immobile and are part of the structural component of the cell? Sulfur, calcium, sulfur, potassium, calcium, magnesium, potassium and magnesium. Yes, question number 5. Any doubt? Sorry, any idea? So structural component. So in your DNA, do we have sulfur? No, but proteins have sulfur and calcium is the oldest. So your answer is option A. So moving forward, question 6. So any mineral ion concentration in tissue that reduces the dry weight of the tissue by about 10% is considered toxic. So please remember the toxicity of magnesium. It inhibits the uptake of calcium ion. Okay, fine. So the process by which mineral is absorbed is called as, so is your mineral absorbed by your passive absorption, active absorption, both A and B or none of the above? So your mineral is absorbed to some extent by passive and then by active, right? So initially it is active because the roots have more compared to the soil and then it is passive. So both A and B is the answer. So question 8, which of the following represents the abiotic mode to convert nitrogen to nitrogen oxides into the soil? So abiotic mode, without your bacteria, which helps in the conversion. So that is lightning offers some power, it is not temperature, it is not ammonification, nitrification. So ammonification and nitrification are by your bacteria, right? Hi Manoj, yes, welcome to my lecture. So please do like, share and subscribe. Okay, so here the answer is lightning. So biological nitrogen fixation. So reduction of nitrogen to ammonia by living organisms is your biological nitrification. So nitrogen to ammonia, because plants can take up in the form of nitrogen. So enzyme nitrogenase is capable of reducing nitrogen. So which organisms have these enzymes? They are known as nitrogen fixers. So nitrogen fixing microbes could be free living or symbiotic. So these are free living, aerobic, anaerobic, free living, and these are cyanobacterium. Okay, so please remember the example. So we have several types of nitrogen fixation. Yes Manoj, good evening. So we can fix nitrogen to the leguminous plants, that is done by your rhizobium, right? Sometimes we can fix nitrogen with the non-leguminous plants as well. Okay, so that is your Frankia. So Frankia helps in fixing nitrogen in non-leguminous plants. Okay, Devisha, now is this session for NEET 2021? Yes, so we are winding off the syllabus. So we are revising all the 3 chapters of plant physiology. Okay, yeah. So this much you have to remember, and the nodule is the formation is pink color because of leg hemoglobin. So you should remember nodule formation like this, and this nodule or this nitrogenase enzyme is sensitive to oxygen. So that is why it is prevented using your leg hemoglobin. Fine? Yes. So what is the amount of ATP? So for 1 molecule of nitrogen it is 8 ATP. So for 2 molecules of nitrogen it is 16 ATP. So leg hemoglobin helps in the transport of food in plants, nitrogen fixation, protecting nitrogenase from oxygen. So just now we discussed this, right? So ion transport in root occurs passively through channels, actively through channels, actively through carrier, through both symplast and apoplast. So what would be the answer here? So how does it occur? So ion transport in roots. So from the soil to the roots it is passive. So ion transport in roots, now it has crossed the soil, now it will move from the root into the deep. So here it is 1, 3 and 4. So here it will be option C. Okay Devisha? Yes? No. Not okay. So even this is right. So symplast and apoplast. So when it moves towards the root in the endodermis, you have symplast and apoplast, right? So now we move into the last chapter, the plant growth and development. The most important part in this chapter is you will remember the 2 graphs, arithmetic and the geometric. Yes. And number 2, you will remember what is photoperiodism, what is vernalization. Yes. So when you talk about photoperiodism, you will remember short day, long day, neutral plants, and you will remember the 5 growth promoters as well as your inhibitors, right? So that is your auxin, glyberulin, cytokine, ethylene and your ABA. So let's quickly solve the 10 questions, so we'll be done for the day. So the topics that we read in this chapter are this, so it might be a bit tricky, so it is a huge chapter. So in arithmetic growth rate, when the length of the organ is plotted against time, the nature of the graph curve will be linear, sigmoidal, parabolic or hyperbolic. So what will be the answer? So in your arithmetic growth rate, so your arithmetic growth rate is like this, right? So here I am plotting against time and the length of the plant. So in a given time, how is the length? So what does this graph represent? So it is a linear growth, right? So it is a constant linear growth. So here your answer will be linear. Fine? No, it is linear. So we are asking the graph. So it is a linear graph in arithmetic, right? So here it is LT is equal to L0 plus RT. So geometric, it is like this. In geometric, it is sigmoidal. In geometric, it is sigmoidal. So here, this is the formula. W1 is equal to W0 plus, W0 e power RT. So hormone responsible for bolting is indole acetic acid, kinetin, ABA and gibberellic acid. So here it is your gibberellic acid that is used for bolting, right? Yeah. So which of the following synthetic phytohormone was discovered as a breakdown product of DNA? Kinetin, 2,4-D, NAA, thidiazuron. So these three are synthetic. So they are asking a phytohormone. So this is kinetin. So this is option A. So how was kinetin discovered? So we understood there is a hormone known as cytokinin. So we know cytokinin also has an effect on cytokinesis. So this helps in cell division. But naturally we could not find any cytokinin. So when we searched for components similar to this, we found out zeatin in plants. So it was present in your corn kernels and in your coconut endosperm. But initially it was discovered as kinetin in your autoclaved herring sperm DNA. Done? Yes, very good. So the highest concentration of kinetin is present in the regions of continuous growth and meristematic region. Meristematic region only mature leaves or ripened fruits. So it is present because it helps in cell division. So cell division is in actively growing regions, right? Yeah. So question 5, senescence in plants can be promoted by applying auxin, cytokinin, gibberellic acid or ethylene. So it is understood it is ethylene because these three are growth promoting, right? So question 6, effect of low temperature which shortens the vegetative period and hastens the flowering period is known as photoperiodism, transpiration, vernalization or photolysis. For flowering here it is vernalization, right? So for which question you are asking? So senescence is by your ethylene. So that is no doubt, right? Yeah. So here hastens the flowering period is your vernalization. Yes, Anthony, you're right. So you should know what is photoperiodism, right? So they give the amount period of light. So that is photoperiodism. So here you have short day plants, long day plants, right? So I want you to remember the examples that is important. Short day plants, they need less photoperiod than the critical level. So who are the examples? You have rice, you have coffee, so you have tobacco, so you have soya bean and you have chrysanthemum. So all these are examples of your short day plants, okay? So then how about your long day plants? So they need more photoperiod than the critical level. So that is long day plants. So example is your wheat, your radish, then you have your cabbage, you have your spinach. All that you don't like but all that is healthy comes under your long day plant. When mummy cooks all the healthy things, we take long time to eat. Remember, long day plants. Okay. Then you have day neutral plants. So these plants, it does not matter if they have more or less, but they grow properly. Okay. So what are the examples that can come here? So here in the day neutral plants, you have sunflower, you have tomato, you have cotton, and you have sunflower. Okay, so these are the examples. Yes, of course the examples are important. Okay, so here you can also add cucumber if you are willing to remember, and you also have peas. So all these are the examples. So please make a note of these examples because it will be useful for you to revise. So these are the examples. Okay, done? Fine. Let's continue. Presence of which of the following compound accelerates abscission of flower and fruits? Acetaldehyde, ethipon, ascorbic acid or zeatin? Yes, question number 7 here. So ethipon, that is ethylene, So it is commercially available as ethipon. So this helps in the absorption, right? So ABA is antagonistic to gibberellic acid, cytokine, ethylene, auxin. So here it is antagonistic, it is opposite to your gibberellic acid, right? Okay. So 6-furfuryl amino purine, 2,4-dichlorophenoxyacetic acid, indole 3-acetic acid are examples respectively of what? Yes? So what is the right answer here? So your 6-furfuryl amino purine is kinetin. So kinetin is a modified form of adenine, which is a purine. So 2,4-D is synthetic auxin and indole 3-acetic acid is a natural auxin. Fine. So the last question for the day. So plants requiring exposure to light for less than the critical period, they are your short day plants. So example of short day plants, so which includes your rice, your soya bean, your coffee, your chrysanthemum and your tobacco. Okay. So with this we come to the end of the chapter. Yeah, long day, short day plants, right? Yeah, so with this we are done revising all the 3 chapters. So now all the 5 chapters of plant kingdom are done and today's combination is done. Okay, yes. So we are just left with 2 more chapters to complete for the syllabus of your NEET. So we are just done, sorry, we are not left to complete, we are left to revise with biotechnology 1 and 2, which will be done in the days of Monday and Tuesday, after which we will have full revision menting. So I will not have full revision menti every day. So that will waste a lot of your time. So if we are getting done with the syllabus on Tuesday, so we will have your full revision menti after 1 day, so that you have a break in between. Okay, so when I finish my portion on Tuesday, your first full revision test will be on Thursday. Okay, so I will give you a gap for 1 day so that you can study, right? Okay, yes, I will give these PDFs. So for that you have to download eCareerPoint app. So download eCareerPoint app. So there you log in with your mail ID, right? So in the down panel you have free stuff. So go to our YouTube channel. So our YouTube channel is 𝐬𝐿𝐴 Medium. So go to biology, go to syllabus wrap up. So you have today's PPT. Okay, done? Yes. So Yamuna Devi, so thank you for attending my lecture quietly. So please be please do like, share and subscribe. Yes, you get the stuff for free, no money, because this is a free platform. So YouTube is not paid, right? So you get the PPTs for free as well. Yes, Manoj, thank you. So this is me wishing you more and more. So have a great weekend. So prepare well. If any doubts, get back to me in Telegram group. I am always ready to get back to you, but please give me some time. I may not get back instantly, but eventually I will, or gradually I will. Okay. So thank you so much, and this is me wishing you more and most. So please take care. Have an extended weekend. Yes. Bye, and I will meet you on Monday. Until then, stay tuned and take care. Bye.